Well, I think in the short term, the most exciting developments are happening in targeted therapies and in immune therapies. And I'll mention in the short term for targeted therapies, I believe Venetoclax targeting BCL2 in multiple myeloma offers great promise. As we know, Venetoclax targets BCL2, and Velcade dexamethasone versus Velcade dexamethasone Venetoclax, the so-called Bellini trial, did show a progression-free survival for the addition of Venetoclax to Velcade and dexamethasone. However, the overall survival was inferior. Further analysis and ongoing studies, however, show that in the 11-14 translocation subset of myeloma and those patients who overexpress BCL2, Venetoclax together with Velcade dex confers a significant progression-free survival advantage. So I believe one of the therapies that will be exciting for us in the very near future is Venetoclax in this subset of patients who either have 11-14 translocation or overexpress BCL2. Now, we've just had an FDA approval of Isotoxumab, a CD38 antibody that has different qualitative mechanisms than daratumumab. It was approved with Isotoxumab pomalidomide dex versus pomalidomide dex based upon a progression-free survival advantage. Time will tell, but it is our hope that Isotoxumab will maintain activity even when daratumumab does not. Daratumumab news is in two different areas. One, the Griffin trial, which added daratumumab to Revlimid Velcade dexamethasone compared to Revlimid Velcade dexamethasone, or RVD, was a positive trial. The progression-free survival was significantly enhanced, and the MRD rate was about 50% in the patients who had daratumumab added to RVD as initial treatment. Very recently, the FDA has approved subcutaneous daratumumab for therapeutic use, and that's newsworthy because for both patients and caregivers alike, that will make daratumumab a much more user-friendly treatment. And then finally, just to mention some of the exciting immune therapies that I think are going to be most exciting. One is the immunotoxin belantamab mafodotin. That has actually shown activity as a single agent in far relapsed myeloma, the so-called pentarefractory patients' myeloma that is refractory to the first and second generation proteasome inhibitors and the first and second generation immunomodulatory drugs, as well as a CD38 antibody. In that setting, the response rate is in the range of 30 to 35%. The side effect of keratopathy can be avoided by modifying the dose and utilizing corticosteroids. So this particular BCMA auristatin immunotoxin called belantamab mafodotin is going forward towards FDA approval. A second exciting BCMA targeted agent class that I think will make a difference are the bispecific T-cell engagers. I'll mention two very quickly. The CC93269, which is the BMS bispecific T-cell engager. It actually has two binding domains to BCMA and one for CD3 on T cells, and presented, at least at the American Society of Hematology, in a phase 1 trial when the dose of 6 to 10 milligrams was achieved. 90% of patients with far advanced myeloma had activity, including 40% MRD negative. There's another bispecific T-cell engager, or so-called BiTE, called AMG 709, AMG 701 from Amgen. It has not been presented in terms of its clinical data, but we and others have studied this preclinically alone, and especially in combination with immunomodulatory drugs. One can get very impressive anti-myeloma activity at very low effector T cell to target myeloma cell ratios, suggesting it will be very active and have a good therapeutic index. And finally, just to talk about CAR T cells, at ASCO this year, there will be an update of the BB2121 experience. As we know, published in the New England Journal previously, far advanced myeloma patients who received 150 million or more of the BB2121 CAR T cells had a response. 16 of these patients had MRD negativity. The issue for CAR T cells, and this and all others, is to make these responses durable, as the progression-free survival was only about 18 months. Now, the CARTITUDE-1 trial from J&J is an extension of the original CAR T cell studies coming out of the Legend Company in China. And their CAR T cell targets BCMA, but has two binding domains for BCMA. The CARTITUDE trial has been presented at the American Society of Hematology and will be updated further this year, but it's also very exciting. 100% of 17 evaluable patients with far advanced myeloma had MRD negativity, and very few patients had relapsed at 6 months follow-up. However, a word of caution, as I mentioned for the BB2121 experience, relapse progression-free survival median was 18 months. So we need more clinical follow-up of the CARTITUDE trial before we can conclude that they have long-acting responses. And I'll just mention in closing that there are some new ideas about CAR T cells, whether they be RNA-based CAR T cells that could be given repeatedly and have transient activity, so-called binary activated T cell with chimeric antigen receptors, or so-called BAT CARs, where you can have a generic CAR, if you will, that could be partnered with any antibody that we currently have, whether that be against solid tumors or liquid tumors. And so we could extend the utility of CAR T cells much more broadly than present. And then peptide-stimulated T cells, where we can actually vaccinate patients against their own myeloma, generate memory T cells, expand them, and use them as we would CAR T cells to transfuse back these peptide-stimulated central memory T cells back to patients. The beauty of this is these T cells are naturally stimulated and so should not cause much in the way of cytokine release syndrome. And secondly, we can then vaccinate patients in order to maintain these central memory cells with anti-myeloma activity. Finally, combinations, I think, are the future. I think, for example, we might want to vaccinate patients and generate in them anti-BCMA central memory T cells, and then use a bispecific T cell engager that would attract these already activated BCMA selective memory cells to the myeloma and perhaps enhance the response, extent and frequency, but in particular, the durability, since these are memory T cells. So honestly, I think the excitement, as I mentioned earlier, is palpable in multiple myeloma. We know about the biology. We can target the tumor in the microenvironment. We know about targeted therapies, and I mentioned Venetoclax as a very exciting one. And we have all of these immune therapies, not to be used alone, but to be used together. And as I mentioned earlier, I really think we can now achieve this MRD negativity with targeted therapies, with or without transplant nowadays. And I think we can use the immune therapies to maintain this MRD status and lead to a potential cure.